Thank you, Dr. Wang. It was a fantastic job to do as the last presentation, and definitely are like in the first rank of the competition. Because you have a completely different approach from everybody else, and as you say, with many unique advantages. And so we've got several different contestants here. No, seriously, we have a lot of very interesting approaches to the question that we've been analyzing, and I'd like to now invite the other distinguished participants up to the microphone. Please announce your name and present your question to everyone that you would like.
Dennis Smith, Clemson University. My question specifically is for Professor Wang, but I guess you all can address this. It's about scalability of your particles, and you touched on that a bit, and I think it was about a 2% yield from the tobacco plants for one of the viruses. 200 grams per 2 kilos, yeah. So 2%. So, oh no, 10%, right, 10%. Anyway. Is there a way to either enrich the virus on the plant and to increase the yield, or, I know nothing about viruses, is there a way to actually incubate replication of these viruses once you've isolated them?
So good question, Dennis. I think the thing is that both ways possible. So we can, by choosing different plants, we can have a better yield of the virus. But when you think about this one, is already a lot. So 1 gram from the 1 kilograms of the wet leaves. So when you think about the 80% of the wet leaf are water, so we really talk about a lot of biomass in it. But for many viruses, like tobacco mosaic virus, we can easily produce in the E. coli. In that case, we can even get a much faster turnover and easy manipulation. So it is possible. Yeah.
Rutledge Spanky, MIT in Hong Kong U. For the TMV, I have several questions, but we'll start with that. Have you looked at putting those into arrays and tried to distribute charge either on the inside or outside as a fuel cell?
No, I haven't done that. But we have done we can change the charge inside and outside. But I'm not really go to that direction for the fuel cell. But I know there are one group is working on the photovoltaic. It's in Berkeley, okay. Matt Francis.
Mm-hmm. I was just thinking you could use it as a capacitor, and you could build up charge either on the inside or outside to isolate the two. This is basically for everybody. We saw respirocytes earlier today. And I didn't ask my question earlier, but about modularity, because the modulus is going to be very high on those things, so it's going to cause some damage. I would like you guys just to speculate on the possibility of putting oxygen in some of the liposome, lactosome type structures, and whether or not they could be used for quick reperfusion after a stroke or an ischemic event to try and limit reperfusion injury after a stroke, and maybe flow those in before you allow blood flow to get oxygen, and then open it up with like TPA or something like that. I know that's a big question, but...
Let me go ahead first. So my collaborator in MUSC, the medical school in South Carolina, University of South Carolina, Xu Jingwen, asked me the exact same question 2 months ago. I don't have an answer for that. Really don't.
That's the short one. Yeah, I agree. I think oxygen is a big challenge for a molecule that small. To be able to hold it inside anything is enormously challenging.
Well, I guess using chemistry is the only answer to bind those oxygen molecules using some kind of oxygen
generator.
Yeah, receptor binder inside some. Well, you can probably bioengineer those molecules inside the viruses.
But I think the key question is they are talking about in this special surgery process, you need this time frame very quick.
Right.
It's really hard. I have done the calculation. It's really difficult for any reaction to do that. I'm not saying it's impossible. I have done that one. So far, I haven't found an efficient reaction to do such kind of supply.
I think conceptually it's possible. If you look at the glove boxes that we are all familiar, the glove boxes have materials that absorb oxygen. And those materials are extremely efficient. I'm not sure what size and what type materials that people use in those glove boxes. So conceptually, you can design material, probably nano-sized materials, incorporating those selected types of inorganic materials which can absorb oxygen. So the next issue would be, how do you ensure that these materials are protected? So one option is to have a polymeric shell, since polymeric materials can diffuse, gases. And then at some point when you want to release your oxygen, I think you could look at probably a chemical mechanism by which you can diffuse out the oxygen. So conceptually, I think it's a possibility.
Yes. It can be done, and I'll show you pictures.
Okay. Then the last question I have, and then we can all go, is have you guys looked at inhalation of some of these for drug delivery as opposed to just putting it in a vein, because you get much better biodistribution if you can do an inhaled version, and these things are very small. Depending on the characteristics of it, you can either get super high adherence or low adherence based on structure, which you could probably design in. Could you guys speculate or talk about some of the work you're doing in that area?
Well, I was last time, so I'll be first this time. Yes, it works. You can the vesicles, you can do that. You can also do small particles, porous particles and hollow particles, which we've done, and that's been done, and it seems to work pretty well. The problem is getting things to release out of them in a controlled fashion where you want them to release and when you want them to release, and that's still something that's under investigation. But getting things, inhalation of small particles, especially charged ones which do not aggregate, is actually not much of a problem.
Just adding to that, actually, this is a very well-researched area using nano-sized polymeric materials. In fact, if you look at the patented literature, I think several polymeric nanomaterials have already been designed for controlled release of drugs through the inhalation process. So, yeah, probably I think Russell is here, so he can answer this question better. He's an expert in polymeric nanomaterials. Any comments, Russell?
I'm sorry, I just walked in a few minutes ago.
Oh, okay. No, I think the question is, can we utilize polymeric nanomaterials for drug delivery through the inhalation process?
So pulmonary or intranasal? Pulmonary is being done. But it's
bad days for pulmonary drug delivery as well. Pfizer just withdrew inhaled insulin in the fall. $8 billion. And Lilly just canceled their inhaled insulin project. So while it's an attractive approach, and we are kind of futurists here, it's bad days at the present.
Well, my personal view on removing those from the market had much more to do with it being a bad product for insulin more than the inability to utilize that route for systemic delivery.
Yeah,
but there simply may be a systemic.
the problem was that they didn't fall apart. They didn't have a problem in terms of getting everything in or any sort of bad side effects. The problem was that they were no more effective than injecting it, okay? And it was actually much easier in this case to inject it for people.
I think the problem was, as you said, they weren't being used.
There was also a patient perception which wasn't supported by quantifiable data. So
talking about the implantable.
And as a
I don't think it's totally
as-
implantable. I mean, they just put a probe underneath your skin, and the Medtronic mini pump is kept in their pocket or something like that.
But as an asthmatic, I can tell you, inhaled therapies work beautifully, okay, 4 times a day.
Yes. I'd like to ask a hypothetical question of the panel, and maybe see what different answer each of your platforms would provide. Suppose there was a small sugar molecule, a small man-made sugar molecule, an amino sugar, which was shown to be effective against all clades of the influenza virus, including the feared bird flu, or H5N1. And it accomplished its magic by basically interfering with viral replication in the endoplasmic reticulum. So this would be a therapy that one could be given after they were infected. It wasn't really a true vaccine. But the problem was when given this amino sugar directly without a carrier, it never made it into the cell. And so my question is, what would be the advantages and weaknesses of each of your platforms in delivering a small, broad-spectrum antiviral, small sugar molecule into the cell? Take into account you have to figure out which cells to go to. They may not all be in one part of the body, and that's really the only constraints. So maybe start with you, Dr. Kumar.
Yeah, actually, if you look at the drug delivery options that we have, it can be very complex, right? But then one needs to apply, depending on the situation, whether do you want to go ahead with a complex solution or a simple solution. So maybe the first approach would be to start with the sugar molecule and identify if there are any unique receptors present on the cell in which you would like the sugar molecule to interact. So once you identify that, the first approach would be to tag that, say, ligand for that specific receptor to the sugar molecule and see how effective it is. And from then onwards, you can increase the complexity of your delivery systems, whether you would like to have it released in a timely manner or whether you would like to have it protected for some time before it's released. So various issues come into picture.
So your basic answer is that you don't even have to go to the point of having it become a cargo inside a liposome or a nanoparticle. Just try to affix the antigen of interest directly to the sugar molecule.
That's right, yeah. So many times simple procedures work very well. Okay. So you don't even need nanotechnology.
Okay, Dr. Deshawn?
Well, I think the major problem you're going to have is, at least from my perspective right now, is targeting, getting it to where you want it to go, because in this, everything's going to be dependent in this case of not having it sort of be non-discriminate in cells. The other problem, if you want to get very high concentrations on the inside of these, you're going to have to conjugate it so that it ultimately has some sort of charge on it, unless to get really high concentrations inside, which is what you would like to have. So remember, volume is everything here. Surface is what to be avoided. Volume is everything. But
okay, great. Dr. Wang?
I wouldn't use my silicon nanotube in this case. I would go with Phil's vesicle, I think, to concentrate those amino sugars in the cells, and that would be a better way, or Dr. Kumar said,
okay,
something like that.
A role for magnetics?
What my talk was primarily ways to trigger the release, and this is more of a challenge of getting things inside the cell. I think Jim Leary's talk was very good on having the different shells that break apart as you cross the cell membrane. So not so much for magnetics for either targeting or probably triggering in this event either.
Okay.
Yeah, I think I have to say one thing is, first, I'm not a big fan of use of virus to deliver the drug. But one of the fundamental things I didn't mention is, actually I mentioned a little bit, is the immunogenicity. The immune response is very strong for the virus. However, saying that, I think for your particular question, use the virus is a really good scaffold. The reason is when you have amino sugar, such kind of molecule, when they try to block the virus replication, is well known the polyvalency is very crucial there. So you do need to have some kind of either dendrimeric scaffold or virus scaffold, how this kind of display to have not only one, is a multiple amino sugar in the very high local concentration. So that can much more efficient to cause to stop that.
Yes, it's like an example of virology helping immunology.
There are actually one work by, I think, Mary Manchester has published one paper about 4 years ago. So he has shown for the measles virus, this replication, you can display a special antigen or special sequence on the copy mosaic virus is 200 times more efficient than the same dose of the small molecule. But in order to targeting, I think that one thing we have to do is just use a layer by layer method to have the virus as the inside this kind of drug is a polyvalent drug, and outside use some kind of polymer or some other thing to grab it for the targeting. So in that case, you can reduce the immune response. In the meantime, you can still use the polyvalency of the virus to do the work. Yeah.
Great. Any other questions for the panel? Everybody's thinking dinner. Okay, so we've had an amazing 11-hour day. I mean, that's not a nano particle day, that's a gigaparticle day. So everybody deserves a great hand of applause for themselves. Thank you very much.